Great. Okay. So this is sort of weird presenting to a phone, and I think whenever I've given this talk before, I guess the most important thing is I'm always available to talk, and there'll be time for questions, and I'm going to try to use no more than 35 minutes to answer the questions. But feel free to contact me, and I'll have information at the end about that. Having spoken to the i5k folks, they want me to sort of focus on sort of three bits, a little bit about me, a little bit about VectorBase, you can see from this introductory slide, and then some sort of experience of the various projects I've been involved in. As the PI at VectorBase, I'm sort of automatically included in a lot of these steering committees, and per the request of Anna and others, I've summarized my opinions. Of course, there are other ways of doing this, but again, if you have any questions about what I present or anything you want to follow up on, I'm always available. I think it's the most important part of the presentation. So with that said, I sort of structured this in sort of three parts that are roughly 15 minutes each. If I was giving this talk in person, I would ask how many people are novices versus intermediates versus experts. I've seen a few of all varieties on the call, so again, hopefully this is useful for you all. I have the luck but also this fortune of being the third one, so a lot of what I do ties with what PacBio presented and the sort of high-C scaffolding folks presented, so I'll sort of skip that and focus more on the BRC, what we do, if you have any questions about that, and then in sort of ode to the PLOS series, my own personal 10 simple rules for Accessible Genome Project, the means to help you guys think, and then also maybe to spur some discussion. So first and foremost, I'm not an arthropod person. Most of my knowledge comes from this book you see here that I got when I was at Kansas State, my son's favorite book we read every night. So I'm a bioinformatician by training. I do have a biology background, but my training actually was in microbial genomics. And then I did my PhD working on the corn genome, and then coming here I've been involved in mosquitoes. And I think what's great about working in arthropods is that for me as a bioinformatician it's very challenging. So a lot of the issues you guys may be thinking of or dealing with we've dealt with before. I try to fold that into later parts of the talk, but most of what I will talk about will be computational, it will be assembly focused, it'll be sort of more about what you can do in terms of assembling that's pretty agnostic. But I'll sort of focus on more on mosquitoes because that's what I do and it's useful there. But again, I'm not an entomologist, but hopefully the things I'll talk about will be useful for you guys as you think about sequencing or analyzing your favorite arthropods. The other thing I will say, and this has sort of changed a little bit, is that in the sort of BRC, so the Bioinformatics Resource Center aspect of it, a lot of what we do is very applied. So we're funded by NIH and NIAID to present resources that ultimately will reduce vector-borne disease transmission. So here's a picture I took about a year ago in the Atlanta airport with a VectorBase picture on it of the Aedes aegypti mosquito. And again, ultimately what we want to do is connect genomes to disease. And a lot of what I'll talk about for the VectorBase portion of the talk will sort of connect there. And again, I'm not trying to necessarily plug VectorBase. I think what's important, and I think what i5k does since I've been involved in the very beginning, is to try to create a more inclusive umbrella of our resources, and if there's things that we're doing that are useful to others, that's great, and we're happy to share that. All of our software and tools are freely available, so again, it should be pretty easy for people to go ahead and use what they see as useful. So I know that there's people who aren't part of VectorBase or know of what's going on, so here's just a brief introduction to the BRC. We're one of 4 or 5 resources funded by NIH, depending on how you count the influenza database. And originally there were 8 that were spawned by the Amerithrax attack on Congress. There were no resources for important pathogens and important sort of bioweapons, and so they decided to create this portfolio of projects that are focused on emerging and re-emerging diseases. And vectors are thrown in there because malaria and dengue and Zika were thought to be something that would be important for the global community. And so we're actually on our 3rd 5-year contract. We're funded through 2019. And what's nice about this, and what I usually get asked at meetings, is not only are we around and successful getting funding to provide support to the community, we've been doing it for a very long time. Now, there's no free lunch. So I think what's unique about VectorBase and the BRC program is that we are federal contracts, and as a consequence of that, we have to do lots of paperwork, which is sort of a little bit annoying. But the idea behind it is that we are obligated and basically contractually obligated to provide service. So unlike Flybase or other resources that are grants, there's pretty strict things we have to provide, and we're very much a service provider for the community. So we hope we do a good job. We hope to hear back from you guys if you're using VectorBase. But that's one thing that separates us as a BRC from other BRCs. Here's a picture of our user community. We get between 1 and 2 million hits per year. It's kind of hard to estimate what our user community is. It's probably between 7,000 to 10,000 scientists, loosely defined. So that would be undergrads, grad students, postdocs, and researchers. As you expect, it's mostly focused on the Midwest and East Coast of the U.S., Europe, and Brazil, although we do have emerging usage from Africa and China. We do about 3 to 4 workshops per year at places that are using VectorBase. So the last 2 were in China and in Africa. We do that whenever we can. But that gives you a sense of sort of our users and where the hotspots are of our community. Here's our website. And again, I'm not so much going to plug in this presentation the BRC. We have about 10 things we're obligated to do that are shown here in these tabs. The really short executive summary is that we are responsible for providing genome data to vector biologists. Most of it is sort of vector focused, but we do have a few things that are important comparatively that we've thrown in there. We are also supposed to integrate data from the community, and one of my personal goals is to make sure that the people who generate the data get as much credit as possible. So wherever we can, we try to plug you guys to do the work to incorporate that. What's nice about VectorBase is that we have the resources to curate and integrate across different projects. So it's sort of a one-stop shop where you come to the resource and you can find and visualize data. And so that'll be sort of a plug for moving things forward. And then sort of the last thing that we're trying to do is move more towards translational or sort of non-genome sequence data. So we have transcriptome data, proteomics data, population biology data I'll talk about in a few minutes. And again, using the genome data to sort of facilitate integrating other types of sort of more either basic science, proteomics and host-pathogen interaction data, or the context of surveillance, looking at where the mosquitoes are found and how they actually adapt to their environment. Again, if you want to email VectorBase, we have a little tab up here that you can go ahead and click on. We'd love to get emails from you. Again, we're a service. So even if you're not a vector biologist and you have any questions, feel free anytime to email me directly or go ahead and email VectorBase and we'll be more than happy to help. We provide tools and services. This is sort of the middle portion of it. As you would expect, most of our users use BLAST. I'll focus in on 2 that I think cross different communities. One is the population biology effort that I'm hoping will be useful and interesting to you guys from other communities. And then also Web Apollo, which we've been involved with since the beginning. I wrote a letter of support for that group, and it's been awesome. I know that i5k also provides a Web Apollo instance for their species. We do other stuff, so Galaxy, for example, you can use Galaxy stuff anywhere. I think what differentiates our resource is that we have all the genomes that you may care about in-house, and latest Annotation just makes it easier to do those kinds of analysis. I was asked by the i5k folks, is it possible to hit buttons and do analysis? I think genome assembly isn't quite there yet, and I'll talk about that towards the end. But we are trying, at least for vector biologists, to build frameworks to do RNA-seq analysis in the BRC. So if you have transcriptome data, RNA-seq data from your favorite insect, to basically hit a button and get pretty good results the first time through our resource. Okay, so the Web Apollo stuff. So we did a bunch of, to use the British term, bespoke things before that I think worked for us, but maybe didn't work for others. What was unique about Web Apollo 1, we started this about 2-3 years ago, was you need to have a separate instance per species, and VectorBase has over 40 genome. So we had at one point I think 20 or 25 separate instances. Now we use Apollo 2, and it's great because we have just one for all of our species, and we can generate reports, and they've been really proactive helping us. We're going to try our best, and maybe it is a plug, to work with Moni and other outreach people at Web Apollo to do more arthropod-centric stuff, and we're actually going to have a workshop here in South Bend in the summer that will help sort of go ahead and do that. That's actually cross-resource. So if you have interest in doing informatics, I'll plug that later, but we are going to organize a workshop here to get a sense of combining forces of all of our resources to help you guys do the cool science part. Here's a slide from Dan Lawson to give you a sense of what the extent of our annotation is. It's not uniform. Some genomes get way more love than others for a variety of reasons. But you can see that most of the changes, I think this is over 2 years since we installed Web Apollo 2, is modifications, but we do get new genes added, we do get merged, and we do get splits. And so this is really great for communities to get involved and be part of the team. If you aren't a genomicist, you know your genes, you've been working on them for your entire career, and so this is a really great opportunity for the leaders of the various efforts to get people involved, and we're working on our software stack to do this more frequently, and our goal is to basically try to update the annotation with this data at least once a year and to make it available as soon as possible. So I think what's great about Web Apollo is that now it's a much more sort of real-time system where we go ahead and get a new gene modification, it's available, people can view it, and then go ahead and incorporate that later into the gene. As I mentioned earlier, we are trying to enable sort of more plug-and-play for RNAseq. So we've actually gone through and pulled in all of the SRA data from GenBank and made them available to the community. Wherever possible, we try to make sure the publication, the authors, all that metadata is there so people get credit for it. This is actually some data from insecticide resistance generated out of France for Aedes aegypti. And so I think it is at the point where we can help people do that. So if you're interested in doing RNAseq, we've seen, at least for our vectors, very similar results with different aligners and different tools. More than happy to follow up with you offline about that, but we are going ahead and doing that in an automated fashion moving forward. This is sort of a busy slide, but hopefully it gives you a sense of sort of how we think about this as a resource. So ultimately what we want to be able to do is sort of integrate or sort of create an easy one-stop shop where the data you guys publish get put into an archive like SRA. We create a node that has all the information about you guys and what you did and as much data as possible about the experiment. And then we use two technologies. We use Apache Solr, which is a search-based framework to facilitate searching and downloading and finding data, which you can sort of see in this view over here, as also providing visualization of co-expressed genes or genes that may be involved in different sort of setups. And so most of our users aren't bioinformaticians, so by being able to find the data sets they care about and be able to visualize that, that's important. And then at the bottom is sort of our traditional use case where we go ahead and look at, these areas here are expressed in RNA-seq data. Those are exons. And so we try to incorporate as much of our next-gen data as possible into the browser that then feeds back into the annotation, and then will then lead to more gene modifications, splits, and joins. So it's pretty inclusive, but that's sort of how we view it, is sort of enabling people to ask questions and be able to find data more easily than going to SRA and looking around on your own. The big push, and I'm hoping this will sort of spawn some discussion and some setup, is on looking at metadata. So we have a new resource we call population biology, which is basically our means of trying to connect genotypes to phenotypes. We were asked by NIAID to do insecticide resistant, so we've been doing that. I think we're not the only site, so if you go ahead and look at the data that we have, most of our data comes from either the Malaria Atlas Project, which is a UK-funded effort, or the President's Malaria Initiative for looking at IR resistance data. But what's interesting about this is that, trying to connect the dots, we have a really good resource for storing variation, and so we sort of see ourselves not being the be-all, end-all surveillance resource, but as a means for people to go ahead and start connecting and finding sort of gene-based markers for things they care about, like IR or other kinds of behaviors important for disease transmission. So that's sort of a pie-in-the-sky, multiple-year kind of project, but through things like the 1,000 Genome Project for Anopheles and other efforts, again, our goal is to try to facilitate as best as possible, facilitating QTL and association and GWAS study in our insects. What's key about here, and why I sort of want to highlight it for people on this call, is the underlying framework actually was built as part of the old GMOD consortium with plant databases. So we built this, it's freely available, you're more than happy to go ahead and use what we've done and go ahead and access it, but a lot of the framework we did was sort of plant and mosquito. I'm expecting it will also be useful for other groups if you find it important to store metadata. Most of our stuff is both temporal and spatial. So we go ahead, and this is traditional surveillance data. I'm sure you can sort of see the species down here. But basically, each of these circles is a sampling site. The colors correspond to the various vectors that were found at that site. And then I'm just sort of zooming in on one here where you can see that this African site has mostly Phenestus in purple and Arabiensis in blue, a little bit of Gambi. And that's important because they're all malaria vectors of various strengths, and then we want to be able to know about that for spraying and for other kinds of interventions that may happen in Africa. And so since we're dealing with a lot of, again, temporal and spatial data, we try to build these views. You can use the search box to go ahead and pull things out based on the catch method or the time or the date or the species. And again, this should be very flexible. So if you have an interest in using this for your own species and you care about temporal and spatial data, our goal is that it will be useful and really available for you guys also to be able to use that framework. And so what we're doing right now is trying to build in views. So here is a sample view. We have an IR view that I'll show you in a minute. It gives you a sense of some of the data people care about in the field. And then we also are trying to incorporate search. So the value isn't so much that you can't analyze your own data on your own. It may be that if you're working in Mali, you may want to look at neighboring countries' data and pull that in to be able to see is there resistance, what the allele is, and if there's any integration into your area. So we're working on this. There are a few other efforts that we're collaborating with pretty closely, and our goal is to try to provide one unified front. So if you create data for us, it'll be useful for all the other resources. And again, try not to reinvent the wheel. Try to sort of spread things around and try to use all of our resources to make it better for modeling and storing metadata in the future. Here's an example. And again, I may not be able to see this through the slides. This is just basically a standard insecticide resistance phenotype where you sort of look at the mortality of insects with the background versus the ones you select. You can go ahead and click on various parts of Asia and Southeast Asia. And the idea is to be able to visualize this and hopefully help not only track resistance, but also to make it clear where resistance may occur, and that may inform spraying. So again, we're NIH funded. We do things that are very basic science oriented for the genome side, but ultimately it has to sort of manifest as sort of the samples, and we're trying our best to sort of make ourselves more amenable to more field researchers who care about the phenotypes and the resistance, and then connect that to, so for example, this sample here with 2 has metadata with resistance, but it also has SNP data or expression data to make that available and sort of try to connect the dots of how the genome is reacting and showing that to our user community. So this is all abstract, and this is sort of older data, but I think it will maybe help you get a sense of what we've done. And I've also chosen this because I was informed, and hopefully this is the case. Some of you are novices. This is my own first exposure to arthropod genomics. And so I still call them M-form and S-form, but they've been renamed. So there's Anopheles coluthi and Anopheles gambi, sensu strictu. And they have different ecologies. And so this is really recent, and in our case, maybe 1 million or 2 million year divergence. If you're in the field and you have a mosquito land on you, you can't tell if it's M or S unless you sequence it. Maybe if you're in a rice paddy like here, maybe it's M-form. If you're sort of off the beaten path, maybe it's an S-form mosquito. And so we wanted to do, I guess what I entered the name about 10 years ago, is try to find genomic markers of what differentiates M and S. And like most incipient speciation with gene flow stories, it's not very simple, but we tried doing it with sequencing. And so we did it in sort of 3 passes. The first pass was to get genomes for M&S. We did that. We went ahead and then did resequencing and did that, and then we're actually looking at even more sampling across Africa in a larger project that I'll talk about. But again, this is sort of an ecological question we had. We came into it, and we wanted to use sequencing to address it. This is a slide from Matt Hahn, one of our collaborators at IU, and Nora Bazansky. And so there are about 4 or 5 different ways of doing this, but classically, you want to find areas that have very high divergence between types, so between M and S, and low diversity within them. And so the easiest way of doing this would be looking at FST. Finding an area that has an FST of 1 is ideal, and do various sort of pre-filtering or sampling things that would make it easier to find that one difference, and then that would be what explains the ecology here. Now that's easier said than done. If you worked in the system, you know that's always great to say. It's harder to actually achieve. What was interesting is that we did this genome scan, and this is maybe about 5 or 6 years ago now, and we published in Science. And the blue areas you see here were shown previously, and again, it's maybe 10-year-old data now, to be involved in speciation, but we found these 2 other peaks that I'm highlighting in circles that we didn't know ahead of time. And just by going ahead and doing sequencing, we were able to find new candidates. This one on 2L actually has been shown to be a well-established insecticide resistance locus, where, and it's this average FST over a window, where M has 1 resistance allele, let's call it A, and S has the other one called T, and this is basically it's fixed. So all M mosquitoes have 1 allele, all S mosquitoes have the other allele, and you can go ahead and look at a single position in the sequencing data and find that that's the allele. What's more interesting to me is that we also had this area down here on 3L that we had no idea what it was doing. And so we went ahead and looked at it, and just a sort of example of how I'm envisioning VectorBase, and this was a lot of work and ended up being a PNAS paper that next year, but going ahead and looking at the sampling across Africa and looking at that allele and seeing what you observe in different places. And so it turns out that in Mali, where that original data was from, there's this allele TEP1RB in green, you see here, where all M mosquitoes had that allele, and then S mosquitoes had 2 alleles but not the green one. And then if you look at Burkina Faso, you see the same thing. If you look at Cameroon, you see a little bit of, Ghana, you see a little bit of green there. What was interesting is that that allele in green actually led to that mosquito being less likely to facilitate malaria transmission in lab settings. Now there's a big caveat there whether or not that happens in the field, but it was sort of a very unexpected result that we found just by going ahead and looking at the sampling across Africa. And so again, our goal is to facilitate this kind of research project through VectorBase, where you can go ahead and find a thing that you care about, go ahead and look at the diversity across Africa, and then get a sense based on metadata if that actually is useful or not. This is, again, a busy slide, but gives you a sense of what we're thinking about doing this. So again, we're using Solar as a search framework, but the idea is to sort of start connecting the dots between variation, so getting VCF files from continent scale sampling in the case of the Anopheles data we have, looking at the metadata, put it into PopBio, and build views that facilitate, again, doing what took us maybe a year or a year and a half of more focused results, and making it available to people to do on their own to sort of find those observations and trends. Okay, so I don't know if the i5k folks are on. I have about 20 more minutes to present. Are things going okay on your end?
Yes, things great. Thank you.
Okay, so at the risk of sort of doing something completely different, and not to plug VectorBase, what I was told, and I love this slide here, if you're starting out genomics, this is what it actually looks like. And so this is actually a misprint. This is a cartoon from the Human Genome Project, what, 15 years ago, 20 years ago now, and how to actually do the interpretation. And so ideally, people like me will build cool tools that you put things in, hit a button, and you get a result. The problem is that the pieces are getting larger, and there's different kinds of pieces, and you don't actually have the box. So as a means to maybe facilitate discussion later on, and also maybe if you want to contact me or others in the community, what I've done is created sort of a node to PLS style my 10 simple rules for a genome sequencing effort. These are my opinions. I'm sure there's more than one way of looking at these, and some of these are probably obvious for anybody who's been involved in science. But the i5k folks said that it would be good to present that, and I say this basically by saying that experience is the best teacher. So a lot of the things that I'm good at because I failed multiple times and learned from them. What's great about this community is that we're all pretty open, and if you ask us questions, we're more than happy to answer that. So by being sort of a relatively, say, dispersed, but we all have our things that we care about, and there's different sort of things why we care about arthropods. But I think the one common thread is that we're all glad to help, and I think this seminar series is a really good example of that happening. So with that said, this is my favorite. Bioinformaticians are not alchemists, okay? And so really the result that I am able to produce and everything I've been involved in is related to how the data is and then the questions. If you have a really good question and you design your experiment and sequencing to be able to answer that question, again, think back to the TEP1RB, right, looking at a transect, looking at things that you know about the biology, then that's great. And I say that because everything I work on cannot be inbred and can't really be colonized. It makes my life miserable. But still, even with the latest, greatest tools that we develop as bioinformaticians, the assembly quality degrades the complexity of the sample. So if you have, if you have to sequence 20 individuals, that will be a worse result than doing 5 individuals. And then I say this sort of acknowledging my own failure. Long reads are better, but they're not a silver bullet, right? So we tried and pretty much failed to try to get Anopheles gambi's Y chromosome with long reads. Now still we published it in a good place. We were able to get something out of it. But the structure of the Y is just not amenable to current technology. So maybe that gets better. Maybe 10x and oxynanopore give us a way to get around that. But things are changing very quickly, and again, by having good data and good questions, you'll be able to get something out of it, even if you aren't able to get a beautiful assembly at the end. And the best way I can illustrate this to you all is with data. So in orange is Anopheles gambi pest, which is probably the most model of our vector-based genomes. A lot of work went into it, a lot of love went into it, a lot of mapping went into it of various ilks. You can see that you have a really great assembly, and you have one scaffold per chromosome, basically, or chromosome arm. And then the one below, the 3 below, are basically the same lab with the same coverage and the same sequencing technique and the same everything. Only a difference between minimus, arabiensis, and fenestis is the colony and the source material. And you can see that you get very varying results of N50, which is a measure of what the piece is that half the genome is in or bigger, just because of the genome content, right? So even doing the best practices and the best strategy and the best assembler and so on, the results even today are still very much focused on how inbred your colony is or how lucky you got with your genome sequence. And so unfortunately for me, or maybe fortunately depending on how you look at it, the one that we care about the most in their name is Anopheles fenestis, and even that's very important for malaria transmission. It's probably the worst assembly here. And so we're trying to do things with PacBio and other techniques to make that better. I have decided not to talk about that here, but if you have any questions or want to follow with me, I'm more than happy to do that. The last one, Anopheles stevensii, has been done multiple times, and this is sort of an old sort of best approach using different methods to sequence it. The N50 isn't great, but it led to a pretty good paper, right? So we sort of tend to get structured on, is my genome contiguous enough? I think it's more important to think about, is my genome good enough? So with that said, rule number 2 is a good genome is a useful genome. Now I realize that utility is variable, but really, even back then, so my first exposure to mosquitoes was with my undergrad mentor, and I've only heard him really curse twice in his life. Once was relates to Java, which is sort of a CS joke. The other one was when he worked on pest and how, basically everything failed when they try to assemble pest. We have some results. So Becca Love has a paper that came out earlier this year using an Illumina library in Niscovar, where she got really good assembly of the genes. I'm not an author on that. I'm on her committee. But that's one way to go ahead and do that. And then I think the corollary in my experience, and I'm happy to be proven wrong, is that because there's so much diversity in the things that we care about, you would think that if I have a decent assembly, I'll sequence more individuals and with PacBio, and then incorporate a hybrid assembly. In our hands, at least, it's always been better just to redo the assembly. So whenever we've done Illumina and PacBio and so on, more often than not, we've just used the PacBio only assembly for our results. And so again, a good example is Anopheles gambiae Y. For all the mosquitoes that we care about, what we try to do, and this is, I think works, is at least for mosquitoes, do single pair mating and then do sibs. So yes, it's not one haplotype, but at least you have at most 4 in those siblings, and that tends to leave a much better result than doing this arbitrary number of individuals. To sort of illustrate that, this is a Vantage plot that surprisingly appeared on the front page of the New York Times about a couple months ago. And so whenever you see this sort of bifurcation, this is a single scaffold of an 80 cell line courtesy of Mark Atomi at UCSF, is where the assembly, even with long reads and even with PacBio helping, where it was still ambiguous because of haplotypes or repeats or so on of what the assembly actually looks like. Ideally, if you're working on mammalian systems like human or mouse, you just get one piece. We can see here, this contig here has 4 or 5 different ways in and out. So again, longer reads help with this. Longer reads have helped with this. But you sort of get these weird haplotypes showing up in your assembly no matter what you do. And then down here, you have really tiny ones, right? Are they useful or not? Who knows? But we sort of go ahead and get something very, very fragmented, even with our best efforts at it. So again, if you want to visualize your assembly, there are more better ways of doing that. This gives you a sense of how much diversity exists, at least in mosquitoes. Rule number 3 is don't be afraid to ask for help. I will speak for myself. I was not an expert, even though I had a background in plant genomics of doing mosquitoes. Mosquitoes are much, much, much harder. And so I think what's great, and I'm not intending to really plug the AGS that's happening this summer at Notre Dame, but I sort of thanks to Sue Brown and others for organizing this. I think it's a really great venue, in my opinion, for people who are doing genomics at various levels. And there's always a full house at the bar. There's always some really good discussions that are happening there. So if you're thinking that you want to get involved in genomics and want some help, feel free to ask us. But I think AGS, which happens every year, it rotates between Notre Dame, Illinois, and Kansas State now, is a good venue. And then also email us. We're here to help. We're funded to help. If you send an email to Flybase or VectorBase or insert your favorite resource, I'd love to hear from you. So if you have any questions, there is no such thing as a bad question, and if we don't know the answer to it, typically we'll be able to help you find who knows the answer to it. Okay, rule number 4, and this is sort of experience and maybe my own experience showing up here, is don't put all your eggs in one basket. This sort of seems to be sort of obvious. The things that work the best in terms of large genome efforts that I've been involved with recently, the way they structured it was to create working groups. I was involved, I think, 3 for the Anopheles project. And each working group had a leader, and they all wrote 1 or 2 pages of supplemental text, had 1 figure at least, and then they did that. And I think what was interesting about that is that, at least for, I'm sort of a little bit of an insider, but when I talk to people about the Anopheles effort we published last year, the 2 cool things were looking at sort of reproductive proteins that Fulmina Catarruscia at Harvard came up with, which was her working group, and then the integration between mosquitoes and Anopheles gambiae. And those themselves are basically a small 1- to 2-page paper we came up with, and they both were published in really good venues. So I think it's good. You don't really know when you get into it what you're going to do. You have some ideas, you have some educated guesses, but those are sort of 2 examples of really good research that we came up with as part of an effort that were sort of nuggets. The next one, and this is for me, so no offense to people who are on the call, I'm a computer scientist. We deal with deadlines. The great thing about doing genomes is everyone's excited. It's a great thing for the community. But at the same time, if we're honest, we're all overcommitted. So what worked well for me and my students in my lab, and I think others, was to have a schedule. There's an investment of time. There's always going to be investment of time for a genome project. But what we did for the 16 genomes effort was we had, I think it was about 10 or 12 weeks in our Basansky organized calls. We had each group present every other week or every 3 weeks. And what was great is that you actually had peer review happening in that process. It gave us a deadline to get things done early. We presented that to our peers, and then we got some useful feedback that then led to a better result. So yes, it was 10 hours of my life that went into it, but I think it was well worth it from my perspective. to go ahead and do that. And again, because we had deadlines and well-established things, you didn't have people going off on their own. It was very, very well organized. Number 6, I should probably have this slide first, is teamwork is everything. Most of what I've done and most of why I'm talking to you is because of others. I've had really great collaborators. Here is a subset. I don't have Nora Bozanski's name up here, but I mean, her and I have been working together for, gosh, 9, 10 years now and have a joint lab meeting at Notre Dame. These are all non-Notre Dame people for the most part. And so, again, these are all things that we would not have been able to do otherwise. Now, I have my own pet projects. I've published things in my own research. But I think, looking at, Mara and Alicia for the M&S, they had their own software from Drosophila that was invaluable for that effort. It was great working with them and learning from them. Matt and I have worked on a few projects together, including the Gamby complex, where he sort of gave his sort of chops on looking at using cool things, population genomics. Scott and Greg and Regalides, Igor, Jake, and so on. So I think this is really great, and I have some examples of at least personally where I've benefited from this. But being a team player and sharing tends to work out much better than sort of being in your own camp. Now the hard part here, and those of you who are senior, you realize this, is that sharing credit is always hard. I think my experience has been, and maybe there's always an exception, that there's always plenty to do in a genome project, and so even though the flagship may have 2 first authors and 3 senior authors, the sort of more focused papers tend to be published in good journal venues than they would otherwise, and then they have less work to do because, again, the genome is there and things have been done in sort of more of a collaborative fashion. So the next 3 are inspired by my kid, and hopefully my cold isn't coming across too much in this webinar. I know that everyone probably has a horror story, but my experience has been, sharing is always the best option. You always get a better product, at least with the first flagship paper, than doing it individually. And other things should be archived. So if you have your genome, making it available to others is always a good idea. In my experience, make sure it gets in the GenBank. And then I think, the sum is always better than the parts in my experience. So we've done a lot of really great work with different groups. Again, authorship can be kind of hairy, but typically there's more than enough to do, and usually you sort of work it out as long as you talk about it and have your concerns up front about that. And so as an example of a really great project that I did very little on, but I thought was really great, is the RNAseq group that was led by Pedro and Ellen, and Raphael interacting with him. And you can see there's tons of people on this list, and then there's been about 4 or 5 other papers that come out of this that are really great. So here's a flagship paper, everyone's involved, very inclusive, they shared all the data, and I think everybody was able to benefit from that in the long run. And then also the work we did in science here, you can go ahead and look at that from, I think it's 2015. All right, so I think we have about 15 minutes left in this presentation. I'm going to leave some time for questions. So I have a few more things to talk about, but I'll try not to go through too fast. I do want to point out that one of the working groups that I was involved in with the 16 genomes looked at synteny. This is just a figure from that paper, and I pointed it out because there were 3 computer scientists that were on that call, and we still work together, and we're still working about 1.5 years, 2 years later on that work. So these things can have legs. They do go beyond the actual effort. It's been great working with them and doing that, and so this is a simple figure we went ahead and did, but it's been really useful for us in terms of our next projects, and I think at least 2 or 3 grants have been submitted on this, and it's been great, I think, for everybody involved. Okay, so I have 3 more rules, and then hopefully have time for questions. There has to be a teacher in the room, too. This is usually a labor of love, and it's entirely thankless. Nora Bozanski did an awesome job for all things I've been involved with. I owe her a lot. But usually what will happen is that somebody has to organize the calls and be the grumpy one and make sure things happen. What's great about this, in my experience, is that usually there's at least 1 or 2 really junior, really good people that want to learn. And they do all the heavy lifting of the paper. So I think that tends to work out really well. Having sort of the organized committee doesn't really do much besides organize calls and then having people who do the work. But I think there has to be somebody there. And if you have any questions, again, I think our community is big enough now that we have people who've done it before and can be more than happy to share their own experience. This is my own here that I'm presenting to you. Number 9, and I guess the original kindergarten things you learned was warm cookies and cold milk. warm food and cold beer are good for you. So I'm a big fan of having a kickoff meeting for any project. The ones I've been involved with have all had this, and it's gone really well. You don't have to go crazy. The last one I had was lunch and a small American beer, right? But I think having an organization meeting or two in your big meeting, if it's ESA, maybe it's the CREET meeting, ASCMH, or so on, that tends to be much better, and people know each other and can go ahead and do that. You can still have your jamboree at the end, but I think an hour spent talking over beer makes much of it much easier later on, particularly with the steering committee. If you don't know people, so some of the things I've been involved with, they're great colleagues and collaborators of mine now, but I didn't know them up front, and so just having that sort of social meet people is invaluable as you get started. And then the last one, and again, I'm happy to answer questions, and this won't be the be-all and end-all for you, is that bigger is usually better. We have data literally right off the press from 10x that we're currently analyzing. We've done 3 genomes with PacBio, which probably is the most of the people that I know of in the arthropod community. I'm more than happy to share that with you guys offline. Our experience is that when you do size-selected PacBio, you get a better result than everything else in local continuity. So the actual pieces you get that aren't scaffolded are larger. And what's nice about that is those pieces tend to work well with optical mapping, so BioNano or whatever you want to do to use your optical map, as well as high-C or other kinds of scaffolding. So when we do our reference genomes, as we call them that, more often than not what we do is we do size selection, we go ahead and do PacBio, and then we then add some other layer on top of that. Now the downside is that you need a ton of DNA to do size selection, and so that's definitely a barrier. There are some efforts going on that maybe they can present at this i5k to make that go faster. You still have to remove residual haplotypes by hand. So I can't speak much about the 80s project, but I was talking to a colleague, and literally I think an inversion assembled in both forms. You had an area that was 95% similar between 2 different haplotypes, and both of them assembled. In the Annotation Gambi S, Pimpirina, there's a well-known inversion in mosquitoes, and again, both of them assembled. So what we're doing, and it's not high throughput, is literally going in by hand and just picking out one haplotype per sample. And so that's the hard part. I mean, yes, you'll get a good assembly if you hit a button, but if you're working on things that are heterozygous, you may get multiple alleles assembling, and then that has to be dealt with. And again, we've done a lot of work with high-C in various kinds. We work with phase genomics, less so with dovetail, but we're probably doing dovetail stuff on a different project this spring. So hopefully there's time for questions, and sorry for going through those really quickly. The key, before I leave it to Anna to ask questions or answer questions, is don't feel shy. So again, I think this will work much better if we can do a Skype and see each other and sort of interact. I'm sure others on the call are also probably happy to help you. Feel free to contact me via email here or Twitter or wherever you want to go ahead and do. But I'd be more than happy to talk with you offline and answer any questions you have here as well as any other time. Thanks.
Wonderful, Scott. Thank you. I did realize, unfortunately, my introduction did get cut off. Our mic wasn't quite working right. So I've just sent a private message to everybody, and you can respond to that, or you can send me an email to send in your questions. And so I'll be monitoring that. And along with that, I just wanted to also ask that if anybody is sharing a line today, if you could just send me a message that tells me how many people are on your line, just to help us keep an idea of how many callers that we've had. So I guess I'll get started with one of the first questions. If you were planning a genome assembly project today with limited resources, how would you decide which method to use given the large number of sequencing technologies? Like you mentioned, there's potentials for hybrid approaches. Do you see one sequencing approach arising as the gold standard for a good reference quality assembly?
So as I try to maybe elaborate, I mean, I think reference quality is sort of a misnomer. I mean, a genome is only as good as you can get out of it, right? So what we're doing, and we actually do in practice, and I guess where I'm spending my money, if that's the best way to answer this, is 10x. I mean, 10x has pros and cons. By being able to sort of pull out the high molecular weight DNA and tagging it and assembling it. In theory, that should work out really well. And I know that Scott Gieb at USDA and Hilo has done some work, and I talked to him about it, where he's gotten some really good results with 10x. We're doing it. I don't want to speak too highly about that because I haven't actually seen the data. And then you also get Oxford Nanopore. I mean, Oxford Nanopore in theory would be able to give you really good assemblies, but they're sort of a little bit behind the curve in terms of the chemistry. That can be fixed and probably would be fixed, and it's true for all the chemistries if we're honest. But those are probably the two most viable. So what I'm doing for one of my funded projects is we're still doing PacBio. It's about 10 times more expensive than doing Illumina, and then moving things forward. And then for our generating multiple references for looking at sort of genome scans, Nora Bazansky has been using Discover and their library. And what's great about that is that you can do that on pretty much any Illumina platform. So if you want to go ahead and do Illumina sequencing, and again, it's not as ideal as looking at the long phase reads from 10x, but it does give you pretty good results for the mosquitoes we've worked on. Okay.
Yeah, and actually, can you provide even a little bit more detail there about how your 10x is going?
So most of the data that we're doing is in the planning phases, and we just got the mosquitoes ready literally yesterday. We had a planning call, and I have a colleague of mine who's done some pilot data on it. I mean, I think it's great. The early data we had with 10x was problematic. It's more of a read cloud, so you didn't really know which was which, and it was useful for scaffolding. And then the newest Chromium platform is supposedly better. There's one site, and I can send this, maybe put it in the notes, that's in Texas, I always forget, but pretty much everybody is using them for doing the sequencing. So there's sort of a bottleneck because you have the sample, and then I guess the biggest bottleneck is you need to get the high molecular weight DNA in the first place, as I know that others, not me, are making that move forward. But again, if you want to get long reads, you have to have high molecular weight DNA, and that tends to work better with certain preps and other preps. So really the secret sauce of doing 10x isn't really the 10x platform. It's really, can you get the DNA in the first place that can go into 10x to be sequenced.
And would you choose a different route if you were struggling with your DNA quality? Would you have a second choice?
I mean, I also work on samplites for probably the most fragmented thing that I've ever dealt with. I mean, I think you have what you have, and these samplites are tiny, and if we had better technology or do individuals. What's nice about the Discover is that you can actually do amplification, so you can actually do relatively off-the-shelf Illumina sequencing of an individual, and that tends to work much better than doing pools. Now, of course, there's always the caveat that you have to do amplification, and that may leave some bias. But again, in terms of getting a usable assembly, that probably is the bare bones, and it's worked well. For getting a reference scale, again, everything we worked on, so for Aedes aegypti, we did everything, and PacBio only won. For our mosquitoes, we've pretty much done everything, and PacBio only won. The problem with PacBio is that it may be replaced by Oxford Nanopore, and it's about 10 times more expensive.
All right.
Now, that said, for a mosquito, which is about 250 million bases, and for overkill coverage, you can probably sequence it for $20,000 to $25,000, which I realize is not zero or not really low, but that's way less than it was even 2 or 3 years ago. So it is coming down. It's just not coming down fast enough to be able to do PacBio of your favorite critter.
Okay. Another question here. Are genomic recombination maps still going to be useful for large-scale assemblies?
We've used them. I think in the vector world, what people have been doing, and I've done this in other contexts, is doing restriction associated, so doing rad sequencing genetic maps, and that's been useful. And having a map is always better than having no map. And I think doing RADseq, we've done it. I think most of the effort though is using that for scaffolding and doesn't quite fix the if their genes are fragmented issue. So if we do PacBio only, we tend to get the genes assembled better, at least the ones that are problematic, and then use the genetic mapping stuff to order and orient them. But all the major mosquitoes have had some kind of mapping effort, usually using RAD, that have helped.
Okay. Yeah, and I'll add to that. I think there's a recently published goat genome paper, or maybe it's still in review, but it's openly available. And they did have a genetic map, but they also had Hi-C data, and I believe it was BioNano data. I don't think it was Dovetail, but you can look at how that interplay, how they used it, where one was better than another. That might be of interest to some of the people that are asking those questions. So let's see. So as you said, every genome is useful for certain things, but do you have any coverage or continuity cutoffs that you use to really distinguish between a draft or a reference assembly, or a cutoff at which you would say that genome annotation or gene annotation is going to go well and you're probably going to get pretty complete gene models?
So I'm probably the most pessimistic person to ask that too. So I mean, we've made do with most of what we've had because that's sort of what has to happen, and some of the vectors are notoriously hard to deal with. I mean, black fly comes to mind as being, you know, a really valiant effort and just really hard to, because you can't colonize it, you can't do anything, it's just field samples. I mean, we tend to use BUSCOs and use that as sort of a qualitative metric to see how many genes are there in the first place. And then we've also done things looking at, you know, are the genes fragmented or not using a reference, but it's not really a sort of a fair thing to do because usually we have a closely related relative, and then we do our assessment based on that. So we're not in sort of no man's land. Usually when we're doing our reference sequences, we can cheat and use something that maybe not very, very close, but at least close enough that we can sort of go do the metrics on it. I mean, I think ultimately if you have a, you know, an N50, just use that as sort of a benchmark people can calculate that's more than 10 kb, you're probably okay. If it's 100 kb, you're probably in, you know, you can use optical mapping realm. If it's greater than 1 megabase, you're in great shape. But again, most things that I've worked on have been in the, you know, if you get 10 to 20 kb and 50s, you're probably in good shape. Great.
And I think we've got time for one more question. What has been your most interesting finding as you've taken some of the original assemblies that you had with mosquitoes and you've moved towards PacBio only assemblies and some of those things, that now that you have all of this additional sequence, have you gained some interesting biological understanding from that additional sequence?
Yeah, so I did some thinking about this, because I think that, I mean, the key is that we have marginally better assemblies with PacBio only than we had with sort of the state-of-the-art Illumina we did. I think the key is that, at least for the 16 genomes, and because the Broad Institute cared about them, they did super fancy long-range Illumina libraries that they said only one person could actually do in their facility, and probably cost $30,000, $40,000 to count actually the effort and time and reagents. And so if you look at the sort of, again, scaffolding continuity of mosquitoes, those are still the best, right? I think what has changed the game is that you can, with enough time and money, use PacBio and get an almost as good answer with less money and less expertise. So, you know, again, it's not quite there yet because, again, you're not getting 20, 30 kb PacBio read sizes yet, but we are getting, again, much better first-pass assemblies of PacBio than we did otherwise. Now, for Egypti, I'm not really following it as much, even though I'm on the steering committee. What they've shown is that, like, you know, the M locus and other genes that are really notoriously, or cuticle proteins or things like that, they're really hard to deal with. Again, the continuity of the assembly isn't meet some magical number relative to the old one, but they're getting much better assemblies of those neighborhoods with PacBio than they were with 10x or with Illumina. So again, it's not like you're getting, you know, magically getting an N51 megabase, but we are getting much better views of these really complicated genes with PacBio than we were before.
Wonderful. All right, so with that, I realize we've reached the end of the hour, and so I really want to thank everyone that called in, and thank you, Scott, for a great presentation and really a great discussion as well. I also want to remind everyone that our next webinar is going to be on February 1st with Dr. Evgeny Zabavnov and Dr. Rob Waterhouse from the OrthoDB group. And so I hope everyone will be able to tune in then, and we will talk with you all again soon. Thank you, and take care.
Thanks, everyone. Take care. Bye.